So once a microbe has found a portal of entry, its job now is to figure out a way to attach to the host, or how to accomplish adhesion. This is a process by which microbes can gain a more stable foothold and perhaps propagate to a point that they cause an infection. They use a number of different mechanisms to bind to the host cells. First of all, there are fimbriae. These are hair-like structures, or pili, that they use to bind on to the cell's surface. Another thing is surface proteins, or adhesive slimes and capsules. Here you can see some capsules. These capsules will produce slime, which helps them stick to a certain surface. In viruses, we see envelope spikes that you can see down here. These envelope spikes allow them to bind specifically with other cells. Pathogens are limited to infecting only the cells to which they can bind, so in essence they're quite specific. Some helminth pathogens use things like suckers or hooks or barbs in order to bind to their host cells. This table gives you a summary of some of the other adhesive properties of microbes, the particular microbe, the disease it causes, and then a different mechanism that it uses to adhere to the other cell. You don't need to commit it to memory, but it'll give you an idea of some of the variety of mechanisms involved. The third step in a microbe becoming established is surviving the host defenses. Most non-resident biota in a particular body site in the host are likely to encounter resistance from a variety of host defenses when they first enter the system. The resistance can be from resident biota themselves, but especially we'll see certain white blood cells called phagocytes. These phagocytes engulf and destroy most entering pathogens. They do this by means of enzymes and antimicrobial chemicals. However, some pathogens produce antiphagocytic factors. These are factors that they can use to avoid phagocytes. Some bacteria are actually able to kill the phagocytes. They use toxins called leukocidins that are toxic to white blood cells. Notable producers of leukocidins are Streptococcus bacteria and Staphylococcus bacteria. Other antiphagocytic factors involve extracellular surface layers. These surface layers could be slime or the capsules that we've previously looked at. This makes it physically difficult for the phagocytes to engulf that particular species. Streptococcus pneumonia, Salmonella, typhi, Neisseria meningitidis, and Cryptococcus neoformans are all great examples of species that use this extracellular surface layer. You can see in this figure how the extracellular surface layer or production of capsules makes it difficult for the phagocyte to engulf the bacteria. Some invading pathogens can even survive inside the phagocytes after they've been ingested. A couple of notable species using this technique are Legionella, Mycobacterium, and then many rickettsias. So once the pathogen has found an entry portal, and has attached to the host and survived the host defenses, now we need to consider how the pathogen's virulence factors contribute to tissue damage. Virulence factors are adaptations that microbes use to invade and establish themselves in the host. The virulence factors have a varied degree of effects. Depending on the virulence factor, we'll have different degrees of tissue damage. This effect varies greatly from microbe to microbe. For example, cold viruses do barely any damage, whereas the HIV virus severely damages tissues and even kills its host. Microorganisms can inflict damage in one of two ways. They either use exoenzymes and toxins, or they cause damage indirectly. In essence, their presence causes an excessive or inappropriate response. This is what happens in most microbial infections. However, it's more interesting for us to look at the mechanism of coenzymes and toxins and how they act. Coenzymes are essentially extracellular enzymes that dissolve the intercellular connections so that they can penetrate through and between cells in order to cause damage to the underlying tissues. Toxins are secreted by bacteria to diffuse to the target cells, their specific target cells, and then these target cells become poisoned and thus disrupted, which is what we see here. Let's take a closer look at how the extracellular enzymes work. These work to break down and inflict damage on tissues or dissolve the host defense barriers. Many pathogenic bacteria, fungi, as well as protozoa secrete these exoenzymes. Examples of these are mucinase. Mucinase digests the protective coating on the mucous membranes. It's a factor in amoebic dysentery. Keratinase digests the principal component of skin and hair, the keratin, and it's secreted by fungi that cause ringworm. Collagenase digests the principal fiber in all connective tissues and is an invasive factor in the Clostridium species as well as certain worms. Hyaluronidase is an enzyme that digests hyaluronic acid. Now, hyaluronic acid is the ground substance that cements animal cells together. It's very, very slippery, and we find it in all the joint capsules. This enzyme is an important virulence factor in Staphylococcus infections, Clostridia, Streptococcus, and the pneumococci infections. Some exoenzymes react with components of the blood. For example, coagulase causes clotting of the blood or plasma. Pathogenic staphylococci use coagulase. Kinases are enzymes produced by Streptococcus and Staphylococcus bacteria. They're respectively called streptokinase and staphylokinase. They do exactly the opposite of coagulase. They'll dissolve fibrin clots, and this expedites their invasion of damaged tissues. Now a quick look at bacterial toxins. Toxins are products of microbes, plants, and some animals, and they're specific chemical products that are poisonous to other organisms. Toxogenicity is the power to produce toxins, and it's generally controlled genetically and is responsible for the adverse effects of a variety of diseases called toxinoses. Toxinoses can be broken down into two categories. Toxemias, which is where the toxin is spread by the blood from the site of infection, like tetanus and diphtheria, or intoxications, which is where the toxin has been ingested, such as in botulism. Toxins are often named according to the specific target of action. For example, we have neurotoxins that infect the nervous system, enterotoxins, which target the digestive system, hemotoxins target the blood, and nephrotoxins target the kidneys. We can also classify toxins according to their origins. They can be exotoxins or endotoxins. Exotoxins are secreted by living bacterial cells into the infected tissue. They have highly specific targets and thus highly specific physiological effects. There are many different varieties of exotoxins. You can see a summary over on the right side here of the differential characteristics of bacterial exotoxins and endotoxins. Now, endotoxins are not actively secreted, but they are shed from the outer membrane. They're given off when the cell wall of a gram-negative bacteria disintegrates. As this disintegrates, the lipopolysaccharide in this membrane is released into the system, and thus it causes a more generalized physiological effect. There is only one endotoxin, and it is lipopolysaccharide. You may recall lipopolysaccharide is part of the outer cell membrane in gram-negative cell walls. Let's pause for a quick concept check. Which of the following is not an exoenzyme? Botulism toxin, keratinase, mucinase, or collagenase? Well, we just had that one. So the one that's not familiar is botulism toxin. And it makes sense because this is a toxin, not an exoenzyme. All of these ending in ASE are enzymes. Now let's take a look at the process of infection and disease. This means how it's established, how it's spread, and some of the pathologic effects. Microbes will eventually settle in a particular target organ and continue to cause damage at that site. In addition to the effects of the enzymes and the toxins, simply multiplication of the pathogen frequently weakens the host tissues in which they reside. The pathogens can also obstruct tubular structures like our blood vessels or lymphatic vessels, the fallopian tubes, and bile ducts. If tissues become so weakened that the accumulated damage leads to cell and tissue death, the condition is called necrosis. Now, viruses don't produce toxins or exoenzymes. They primarily cause their damaging effect by multiplying and lysing the cells within which they reside. Many of the cytopathic effects come from impaired metabolism and death of the cells. When we look at the progress of an infection, we have four general periods: the incubation period, the prodromal period, the invasion period, and the convalescence period. You can see in this figure over on the right-hand side the progress from one phase to the other and how they occur over time. The incubation period is the time from the initial contact with the infectious agent, or the time that it found a portal of entry, to the appearance of the very first symptoms. These earliest notable symptoms may be a vague feeling of discomfort. This is when we enter the prodromal phase, so just the vague feeling of discomfort. However, once we get into the height of the infection, we're looking at the period invasion. This is where the microbe is multiplying at its highest levels. It exhibits its greatest toxicity here and becomes well established in the target tissue. This period is often marked by fever and other much more prominent, more specific signs and symptoms, depending upon the infection that we're dealing with. Convalescence is the period of recovery. Depending on the infection, each phase could be extremely varied in length. For example, the incubation period could last from hours to years. Many infections do not remain localized, so let's look at a variety of different patterns of infection. In a localized infection, the pathogen is restricted to a specific site. This might be a boil or something on the surface of our skin. Many infections do not remain localized. In fact, spreading is necessary for pathogens like rabies and hepatitis A. The target tissues in these cases are distant from the site of entry, and so they need to travel to their new sites. These are systemic infections. Many viral and fungal diseases cause systemic infections. Next, we'll consider focal infections. Focal infections are initially a local infection. However, a piece of that infection breaks loose and is carried through the system to other tissues. That can be through the blood or through cerebral spinal fluid, and it can be carried to many other tissues. Next, we look at mixed infection, or they can otherwise be called synergistic infection. This is where the same site is infected with multiple microbes. Often they work synergistically, where the microbes cooperate in breaking down the tissues. Often these are called polymicrobial infections. An example of this is dental caries. Then we have primary or secondary infections. In primary-secondary infection, the initial infection is complicated by a second one in either the same or a different location. And of course, infections can be acute or chronic. Acute means that they come on quickly and stay for a short amount of time, versus chronic, they stay around for an extended period of time. So this figure summarizes the pathway a microbe follows when it causes disease. Essentially, a summary of how it becomes established. First, we need to find a portal of entry. We know this can be through the skin, the GI tract, the respiratory tract, the urogenital tract, or from endogenous biota that's gone out of control. Then we need to have attachment. Attachment happens with fimbriae, capsules, surface proteins, or viral spikes. Next, we discovered how they survive the host defenses. They can avoid phagocytosis, they can avoid death by living inside a phagocyte, or there can be absence in the host of specific immunity. So once an invading pathogen has accomplished all three of these difficult steps, it may be able to cause disease. This disease occurs by direct damage through toxins and/or exoenzymes, or by indirect damage, including inappropriate or excessive host responses to their presence. Finally, we'll move on to how the microbes exit the host. This topic we'll cover in our next section.